Am I audible now? Okay. So the chapter that we're going to be doing today is genetic control, right? And this is in AS 9700 and A levels biology, of course. And this chapter is very important because this chapter marks the base of your genetics, and it's gonna be coming in in 3 to 4 different chapters. So you need to make your base in this chapter. Before starting this chapter, I really need to make basic knowledge of your regarding the genetics, right? So genetics is basically the code for storing information. Now, why do we store this information? So, the information is stored for the synthesis of protein, right? So there are going to be 3 to 4 classes for this chapter. I mean, 3 to 4 hours of lectures for only this chapter. And the first chapter, the first class that I'm going to be taking today is going to be the basic class, right? I'm going to be teaching you all of the basic things that you will need in this chapter so that you may excel in the further classes, right? So, synthesis of protein and regulation. These two things are stored in form of codes that are the codes are stored in genetic form, right? So genes store codes. The codes are for the synthesis of protein and the regulation of the gene themselves, right? So in biological molecules, I taught you that the basic structure of protein is amino acids, and amino acids are connected with each other, forming a protein that have different kinds of structure: primary, tertiary, primary, secondary, tertiary, and quaternary, right? So, the primary structure of protein is based upon the sequence of amino acids. So, let's say we have methionine, leucine, and again methionine and different kinds of amino acid sequences. The specificity of the sequence defines the function of protein and the structure of protein. Obviously, if there's gonna be structural protein for, let's say, there gonna be a repeated sequence of amino acids in it, right? And if it's going to be functional, and obviously if structural proteins are secondary in structure, they have hydrogen bonds between themselves. But the core idea that I want to give you guys is that the sequence, different sequence of amino acids is required for the production of protein, just similar to the alphabets, right? So there are different kind of alphabets in English language, right? And you need a specific set of alphabets to make a word, right? So let's say we make biology, right? This is a word, and this has a meaning. So in order for you to make biology, you need to have B from the letter B from English, the I, the O, then L, the O, the G, and then Y. Here we have O as repetition, right? So similarly, there are 20 amino acids that in sequence, just like alphabets in English, are connected with each other to form a protein. Now, this is the thing that I wanted you to explain. Now, how would we know that the sequence of amino acid that we have to connect in order to make a certain protein, right? So, different kind of amino acids bonds with each other, similar to the alphabets that we write in specific sequence in order to make a word that has a meaning, right? So in order to make a functional protein, you need to join specific sequence of amino acids. So methionine, glycine, isoleucine, methionine, glycine, and alanine, right? These are one, two, three, four, five, six different kinds of amino acids that I've six amino acids that I've used, right? I have repeated two in it, right? Two in it, right? That is methionine. So I've repeated methionine and glycine, right? Similar to the O in biology, but this makes a functional protein, right? So this is what I want to explain. And now, in order for you to make this protein, you need to have specific information that instructs the formation of this protein. Like, how is gonna body know that you have to attach M with G, then I, then M? There has to be a certain set of codes. And for that code to execute, you need a place that it is stored. So the place that the code, this code is stored, is gonna be the DNA or RNA. Okay, so DNA, right? And the RNA. Now, there are two things that I want to say. I'm not going to be discussing RNA right now because it's going to be discussed later on when I move towards the protein formation. But specifically talking about the storage of the codes, DNA is used, right? Now, moving on to the structure of DNA. So DNA structure is, let me just draw it. So the basic structure that you need to know right now is carbon sugar, hold on, carbon sugar, and let me just draw it here. So yeah, so the basic structure that you need to understand right now is gonna be this. So this is carbon sugar, pentose sugar, 5 carbon sugar, right? This here we have is phosphate group. And this here we have is nitrogenous base. Okay, so number one, we have carbon sugar or pentose sugar. Number two, we have is the phosphate group. And this we have is the nitrogenous base. Now, in every nucleotide, two of the things are going to be same, right? This pentose sugar and the phosphate group. These two things are going to be same in every molecule of the DNA that is inside nucleus or inside genes, right? So, but the difference comes in this variable nitrogenous bases. Now, let me just draw the 4 of them specifically, each one by one. So, this structure that we have here is, let me just remove these two, right? Okay, so the variable structure is gonna be these nitrogenous bases. there are four types, right? And this that we have here is going to be same in every molecule of the DNA inside nucleus that is genetic, that is used for gene regulation or genetic control, right? So this is phosphate head. I have drawn a molecular diagram of it, and this is pentose sugar. These two things are going to be similar inside the DNA, because this is very much asked in the MCQs. That's why I'm telling it again and again that these two things are same. The difference comes in the nitrogenous bases. Now, let me just draw the nitrogenous bases here. So the difference that you can identify is that two of them has two rings and two of them has single rings, right? So the double ringed nitrogenous bases, the nucleotides that contain two rings of the nitrogenous base, are going to be named purine. So these two are purines, basically. And these two are pyrimidines, right? So let me just repeat it a bit, right? So I explained why we need to store the information and stuff like that. Then I explained the structure of nucleotides. So DNA is made from nucleotide, just like the protein is made from amino acid. The structure, the basic structure of nucleotide was this. The common thing in every nucleotide was this: the carbon, sugar, and phosphate head. And the nitrogenous base is variable in the nucleotides. There are four kinds. Two of them are purine, two of them are pyrimidine, right? Now, one of them is adenine, and second of it is guanine. This is cytosine, and this is thiamine, right? Now, adenine and guanine, you don't need to memorize the structural differences inside the nitrogenous bases, but you need to remember that there are two rings. That means it's purine, and there are two types of purine, that is adenine and guanine. So let me write it: A and G. And pyrimidine have single ring, that is C cytosine and T thiamine. Okay,